Welcome back to our series on the evidence of evolution. In this episode, we'll explore one of the most powerful and modern sources of evidence, molecular biology. While fossils, anatomy, and biogeography reveal how life has changed over time, molecular biology allows us to see evolution at the deepest level, inside the molecules of life itself. By studying DNA, RNA, and proteins, scientists can trace evolutionary relationships among all living things and even estimate when different species diverged from common ancestors. What is molecular biology? Molecular biology is the study of the chemical compounds and processes that make up living organisms. The key molecules in evolution are DNA and proteins. DNA carries an organism's genetic instructions. The genome is an organism's complete set of DNA, including all of its genes. Proteins are macromolecules built according to DNA's instructions, carry out most of the cell's functions, and shape an organism's physical traits. When we compare the DNA or proteins of different organisms, we can determine how closely related they are. The more similar their genetic codes are, the more recently they shared a common ancestor. The analysis of proteins and genomes from both living organisms and fossils provides strong evidence for evolutionary relationships and processes. In many cases, molecular data have confirmed what fossil evidence suggested, and, in some cases, even refined it by revealing relationships that fossils alone could not show. For example, molecular studies helped scientists confirm that birds are the modern descendants of theropod dinosaurs, based on genetic and protein similarities that complement fossil findings. One striking example of molecular evidence is the BMP4 gene. BMP4 is found across a wide range of animals, including mammals, birds, and fish, showing that it originated very early in evolutionary history. This provides evidence for common ancestry among these groups. But BMP4 doesn't just show ancient relationships. It also reveals how small molecular changes can lead to recent evolutionary differences. In Darwin's finches from the Galapagos Islands, DNA analysis revealed that the expression of BMP4 during embryo development influences beak shape. When BMP4 is strongly expressed, it causes the beak to grow deeper and wider, which is ideal for cracking hard seeds. This is seen in the large ground finch. In other finches, the gene encodes the protein calmodulin, which makes the beak long and thin. This gene is most strongly expressed in the large cactus finch. These discoveries show that evolutionary change can occur through variations in the regulation of existing genes, not just through the appearance of entirely new ones. Now, let us examine the DNA similarities. All living things share the same genetic code, meaning that DNA in bacteria, plants, and humans is written in the same molecular language, with the same four bases, A, T, C, and G. This universal code is one of the most powerful proofs of common ancestry. Comparing DNA sequences between species reveals how closely they are related. Humans and chimpanzees share about 98 to 99% of their DNA. Humans and mice share roughly 85%. Even humans and fruit flies share many of the same developmental genes, such as Hox genes, which control body patterning. The greater the genetic similarity, the more recent the shared ancestor, just like members of a family tree. Now, let us focus on protein comparisons and molecular clocks. Proteins, which are made from DNA instructions, also provide a record of evolution. By comparing protein sequences, scientists can measure how much change has occurred over time. For example, the protein cytochrome C, essential for energy production in cells, is nearly identical in all living organisms. Humans and chimpanzees have the same cytochrome C sequence, while differences between humans and more distant species, such as yeast, are greater. This gradual accumulation of differences can be used as a molecular clock, a way to estimate how long ago two species diverged. Knowing the average rate of molecular change, scientists can estimate the approximate time of evolutionary splits, even when fossils are scarce. Sometimes, scientists can extract and analyze ancient DNA from fossils or preserved remains, providing direct molecular evidence of evolution. For instance, DNA recovered from Neanderthal fossils shows that modern humans share a small percentage of their genes with Neanderthals, confirming interbreeding between the two species. Molecular studies of woolly mammoth DNA show how they are closely related to modern elephants, helping reconstruct their evolutionary lineage. Even when DNA itself is too degraded, biochemical traces, like preserved proteins or lipid molecules, can provide information about ancient life forms and their evolutionary connections. Another important molecule to consider is mitochondrial DNA. Yes, mitochondria contain DNA, the only site of DNA outside the nucleus. In humans, mitochondrial DNA forms a double-stranded circular molecule containing about 15,000 to 17,000 base pairs, which encodes 37 genes. In sexual reproduction, it is only the ovum that contributes mitochondria to the zygote. Sperms do contain a few mitochondria, but they do not survive after fertilization. Therefore, mitochondrial DNA is inherited from the mother and passed down through the maternal line only. It does not undergo meiosis and only changes from generation to generation through random mutation. Gene mutations in mitochondria happen faster than in nuclear DNA, because their mutation rate remains steady. Mitochondrial gene mutations can serve as a molecular clock. Comparing mitochondrial DNA sequences across different species helps determine the evolutionary relationships and estimate when evolutionary lines diverged. Another molecule of interest is Y chromosome DNA. The Y chromosome is found only in males. In humans, it is a short chromosome with about 50 million bases and contains 86 genes. About 95% of a Y chromosome remains unchanged because only a small part, the very tips of both the X and Y chromosomes, undergoes crossover during meiosis. Therefore, the Y chromosome is passed on from father to son. Gene mutations in the Y chromosome are now commonly used to provide evidence for relationships and lineages, especially in human evolution. Molecular evidence reveals that a shared genetic heritage connects all life on Earth. From bacteria to humans, every organism uses DNA and RNA to store and transmit information. Many fundamental genes, such as those that control cell division or metabolism, are remarkably conserved across all species. The molecules inside every cell, from ancient microbes to modern humans, carry the signatures of evolution. They tell us that all life on Earth is part of one great family, connected through time by the shared language of DNA. Thanks for watching this episode on the evidence of evolution. See you soon with another educational video. Until then, happy learning.